Welcome to EPG Pathshala. Today we are going to discuss restriction enzyme mapping. Restriction enzyme mapping is a powerful technique today. It is based on the restriction enzyme which is isolated from the bacteria. There are hundreds and hundreds of restriction enzymes isolated today which cleave the DNA fragments, double-stranded DNA fragments, giving rise to fragments of DNA of varying sizes. Restriction enzymes and the polymorphism associated with restriction enzyme, that is the Rflp, have resulted into an advancement in this new technique of DNA mapping today. Since the rate at which the DNA molecules move through the agarose during gel electrophoresis is depend on the size of the DNA fragment, the relative length of this DNA fragment can be ascertained, and this information helps us to identify the exact location of the restriction site. Comparative analysis of the fragments generated by more than 2 different types of restriction enzymes helps the molecular biologists to determine the relative positioning of DNA fragments. The distance between the restriction enzyme sites can be determined by the patterns of the fragments that is formed after the DNA fragment is digested with the restriction enzymes. Restriction mapping has wide applications. This includes DNA fingerprinting, detections of genetic defect, sequencing of the human genome, and in the evolutionary studies. Although the students readily appreciate this new technology, they find it difficult to understand the technique and the logic behind. The experiment and the mapping exercises described below will introduce the students to the methods used in the enzyme restriction technology of DNA. After studying this module, you shall be able to understand first the role of restriction endonuclease in bacterial defense against bacteriophages. Second, the discovery, types, nomenclature and properties of restriction enzymes. Third, how restriction enzymes can be employed in genetic mapping using Rflp as well as the physical mapping, restriction mapping of enzymes. Fourth, how restriction maps are created, both linear and circular. Fifth, restriction fragment length polymorphisms or Rflp and their application. Restriction enzymes, the charioteers of modern technology. Restriction endonucleases are a specialized class of hydrolases that cleave the DNA at specific phosphodiester bond and acts as a defense system of bacteria, restricting the entry of the bacteriophages. Historical perspective. Independently, in 1953, Luria and Human and Bertani and Weigel first reported the phenomenon of restriction and modification. In 1962, Werner Arber and colleagues observed the existence of defense by E. coli K12 strand against bacteriophage lambda, which he digested was possible by bacteria cleaving the DNA of the phage. Surviving phage undergo modification that rendered their DNA resistant to restriction, which is brought about by methylation of DNA. In 1973, Hamilton Smith and his colleague successfully purified the enzyme HIND2 while working with Haemophilus influenza and prophase P22. In 1969, Daniel Nathan prepared the first restriction map by digesting viral DNA SB40 with different restriction enzymes and analyzed the fragment obtained. In 1978, Werner Arber, Dan Nathans and Hamilton Smith jointly received Nobel Prize for the discovery of restriction enzymes and their application to problems of molecular genetics. Types and nomenclature of restriction endonucleases. Smith and Nathans in 1973 proposed the nomenclature scheme of restriction enzymes according to their schemes. First, the first letter of the genus and the first 2 letters of the species name of the bacteria of origin are used to generate a 3-letter acronym. The acronym is written in italics. The 3 letters acronym is followed by an alphabet indicating the strain of bacteria and a Roman number indicating the serotype. Thus, the name Hin2 indicates that it is isolated from Haemophilus influenza serotype D. Restriction modification enzymes were indicated with a prefix Rm. The restriction enzymes cut the sites are indicated by the symbol /. With the discovery of more restriction enzymes, the original nomenclature scheme was revised with identical 3-letter acronym. According to the new scheme, first, the 3-letter acronym is no longer italicized. Second, the restriction enzymes names do not include a space between the main acronym and the Roman numeral. Third, the convention of naming Rm enzyme with a prefix M, R, etc. has been expanded to include the related gene such as controlling protein. Example, CBAM-H1. And the nicking enzymes that cleave G/T mismatch, example V.H-PA2 for the VSR like enzyme associated with the HPA2 system, and N.BST-NBI for a regular nicking enzyme. Properties of restriction endonucleases. Most restriction enzymes have at least 2 subunits. First, a restriction endonuclease for the cleavage of intruding double-stranded DNA, and second, a methyl transferase for modification for protection of the host cell genome from cleavage. Restriction enzymes are broadly classified into 4 different types based on their recognition sequence, subunit composition, cleavage position, and cofactors requirement. Type 1 enzymes cleave at site distant from the recognition site. These are multifunctional with both restriction and methylase activity and require both ATP and S-adenosyl L-methionine for their activity. Type 2 enzyme cleave within or at short specific distance from recognition site. Most require magnesium. Restriction units of this enzyme is independent of methylase. Type 3 enzyme cleaved at the sites or a short distance from the recognition site require ATP but do not hydrolyze it. S-adenosyl methionine stimulates the reaction but is not required. Exists as a part of the complex with a modification of methylase. Type 4 enzyme acts on the methylated DNA. This is the table showing the characteristics of some restriction enzymes where the site of the cleavage of 2 DNA strands is shown by the symbol / and X representing the purine, Y the pyrimidine, and N representing any base. Most type 2 restriction endonucleases recognize and cut at the 2-fold axis of rotational symmetry, that is palindrome, as these read in 5' to 3' direction on both the strands, generating fragments with protruding 5' termini, that is the 5' overhang, known as sticky or cohesive end. The figure 2 shows the DNA digestion with EcoR1 producing the staggered ends. While other enzymes such as SMA1 do not generate a protruding ends but produce blunt or a flushed end. Figure 3 shows the DNA digestion with SMA producing blunt ends. Restriction mapping. The inheritance of the Rflps through different generations provide information about co-inheritance and thus can help in drawing linkage maps, which is also called as the genetic map. It provides description of the number, order, and distance between restriction endonucleoside for the given DNA. The distance between various restriction enzyme sites in the map is expressed in the base pair or kilobase pairs. restriction enzymes digest the DNA molecule and the digested products are separated using agarose gel electrophoresis, and these are compared to the size along with the simultaneously run standard. Double digestion, that is using two different restriction enzyme, is used for relative positioning of different restriction enzyme sites on a given piece of DNA. In doing so, we can measure the distance between the location of the restrict sites. Data from several digestion can be collated to obtain a more informative restriction map. Generating a restriction map on a given DNA molecule or a data. The construction of a restriction map can be elucidated using a hypothetical data as shown in the table and a figure below. Suppose a linear DNA of 10 kilo base pair is digested with enzyme EcoR1 and the resulting fragments are separated by gel electrophoresis, which yield 3 bands of 2, 3 and 5 kb. The same DNA is digested with another enzyme HIND2, which yield fragment of 7 and 3 kb. For map construction, the crucial step is the simultaneous double digestion with both enzymes, which yield 4 fragments 1, 2, 3 and 4 kb. The inference we draw from the experiment is that 7 kb HIND2 fragment is cleaved by EcoR1 into 4 and 3 kb. That is as 4 plus 3 adds to 7, and 3 kb HIND2 is cleaved into 1 and 2 kb by EcoR1, that is 1 plus 2 adds up to 3 kb fragment by the EcoR1. Henceforth, the 2 EcoR1 cleavage site can thus be placed on HIND2 fragment with respect to each other, generating a restriction map. Such maps can be strengthened by information about restriction digestion of same DNA with other restriction endonucleases. Table 2 shows the size of the fragment generating from the restriction digestion of the given DNA, and figure 4 shows the construction of the restriction map of a given DNA molecule, which is a linear DNA molecule. Construction of the restriction map of a given circular DNA molecule. Similar restriction map can be constructed using different enzymes for circular DNA molecules such as plasmid bacterial chromosomes. Using the same data for single enzyme restriction digestion of a plasmid DNA as shown in the figure below, we have the HIND2 would cut the plasmid twice to produce 2 fragments of 7 and 3 kb. Similarly, EcoR1 should be having 3 cleavage site to produce 3 fragments of 2 kb, 3 kb and 5 kb. Double digestion in this case would not yield the same fragment as those produced in the linear DNA. If 7 kb HIND2 fragments is cleaved into 2 fragments of 4 kb and 3 kb here, EcoR1 should cut in between the fragments of 7 kb and 3 kb to ultimately give fragments containing 1, 2 and 3 kb. The restriction map of complete circular E. coli chromosomes having 4.6 million base pair was completed in 1987 using 6 different enzymes that recognize 6 base sequences. Figure 5 shows the construction of the restriction map of the given DNA molecule, which is a circular one. Restriction fragment length polymorphism. Restriction fragment length polymorphism, pronounced as Rflp, refers to inherited differences in individuals due to variation in length of restriction fragment. First genetic map of the human genome was illustrated by Doniz-Keller et al. In 1987. By testing in 1680 clones from the Phage library of the human genomic DNA to detect Rflp by hybridization to certain blots of the DNA from 5 unrelated individuals. Each individual DNA was digested with 6 to 9 restriction enzymes and observed nearly 400 Rflps distributed along the human genome with an average distance of 10 cm. In 1984, Alex Zeffre used Rflp based technique for discriminating among individuals and species. The analysis of the Rflp arising due to the array of the tandems repeats localized mostly in the non-coding region known as the variable number of tandem repeats or VNTR was used and found that the number of the repeats units in VNTR may range from 9 to 100. The variation in copy number of repeat unit between two restriction enzyme sites can give rise to Rflp. Figure 6 shows the evolution of the restriction site echo R1 due to a single base change leads to an Rflp between of individual 1 and individual 2. Techniques for Rflp detection. Detection of Rflp involves following steps which are elucidated in figure 7 below also. First, DNA isolation or extraction. Second, fragmentation of given sample of DNA with chosen restriction enzyme. Third, fractionation of resulting fragment by agarose gel electrophoresis. Fourth, transfer of DNA fragment from agarose gel to a nylon or nitrocellulose membrane by sudden blotting. Fourth, nucleic acid hybridization with probe, that is tagging with radioactivity or an enzyme specific to the requisite subset of the specific DNA. Sixth, determination of pattern of probed hybridization by autoradiography or using chromogenic substrate. Figure 7 shows the technique followed for Rflp detections, which shows first step extraction, second step restriction enzymes digesting the DNA, third electrophoresis, fourth transfer of the membrane, and fifth incubation with level probe, and ultimately in the sixth step we use the X-ray for detection of the different fragments of the DNA. Application of Rflp. Rflp being widely distributed throughout the genome offers several advantages in molecular analysis of DNA by just analysis a small amount of the DNA, that is from a tissue or a blood sample. Rflp have provided valuable information in many areas of biology, including one, Rflp as a genetic mapping marker. It is the first hypervariable polymorphic marker that was used to create the first human genome genetic map. Overlapping Rflps were aligned to generate a linkage map. The linkage map of the human genome was constructed on the basis of the pattern of inheritance of 403 polymorphic loci, including 393 Rflps in the panel of DNAs from 21 3-generation families. Second, Rflp as a genetic disease marker. Rflps associated with a base mutation causing a disease may be exploited for detection of potentially deleterious gene. Example, in sickle cell anemia, substitution of the base adenine for thiamine in codon 6 of beta globin gene, resulting in the replacement of the glutamine by valine. Figure 8 below depict the pedigree and Rflp analysis of a family where a normal and a carrier and an afflicted child is born in a heterozygous parents. Hence, figure 8 we see the pedigree analysis which can be collated with the Rflp profiling and used for a disease diagnosis. Figure 9 illustrates an example of the linkage of an Rflp locus morph1 to a dominant human disease allele denoted by D. DNA at this locus is cut into two fragments with a specific restriction enzyme morph1. Abolition of the restriction enzyme site generate the second Rflp from MOF2 that produce a single fragment on autoradiogram. Rflp analysis on heterozygous afflicted parent DNA shows 3 bands representing 2 different alleles for MOF1 and MOF2. Owing to linkage, future generation of the Rflp MOF1 individuals are predicted to have a high chance of inheriting the disease allele D. Third, Rflp in DNA fingerprinting. Variation in the number of tandem repeats, that is VNTR, of a short DNA segment can be used to generate individual specific pattern called DNA fingerprinting, which may be used in forensics for paternity testing for providing evidences to establish the innocence or the probability of the guilt of a crime suspect. Fourth, mapping and cloning of a gene when the gene produced is unknown. Correct identification of the target gene located and its associated Rflp helps in cloning and the target DNA by walking down the chromosome, that is chromosome walking, from one microsatellite or Rflp marker to another markers flanking the genes of interest. Figure 9 shows the Rflp loci linked to the disease locus that can be used for disease diagnosis. Terminal restriction fragment length polymorphism or TRFLP. The technique employs extraction of the total DNA, the microbial community, followed by the PCR amplification of 16S rRNA gene with fluorescently labeled primers. This PCR product is subjected to restriction digestion to generate fluorescently labeled terminal restriction fragment. The labeled fragments are detected with laser introduced fluorescent detector. TRFLP is used extensively as a high throughput approach for culture independent analysis of microbial communities. In 6th point, Rflp is used in evolutionary biology. Total number of the Rflp differences between different populations or related species can be used to predict genetic divergence among them. Hence, Rflp may play an important role in the studies of evolution. Let us now discuss the summary for this module. I hope by now you should be able to understand the principle and the step involved construction of restriction map. The ability of the restriction enzyme to cleave the DNA at a specific site is the cornerstone for this particular technique. The nomenclature of the enzyme is based on a simple model that is introduced by Smith and Nathans. The name of the enzyme tells us about the origin of the enzyme, but does not tell us about the cleavage site of the enzyme that has to be determined for each individual enzyme. The recognition site for most of the enzyme is a palindromic sequence, usually of either 4, 5 or a 6 base pair, such as AGT for ALU1 and GAATTC for EcoR1. The cleavage sites of an enzyme falls into 3 categories. In one, they have either flused or a blunt end. Example, flused or blunt end in which the recognition site is in the middle of the sequence, and the other 2 can be 5' overhang or a 3' overhang. The activities described above is designed to enhance your understanding and retention by illustrating the structure of the DNA, restriction and digestion site of an DNA. The activities described above is designed to make you understand and retain by illustrating the structure of the DNA, the sequence at which the restriction digestion of the DNA, analysis of the digested DNA by gel electrophoresis, and the principle involved in construction of the DNA genetic map from the fragments digested. The differences in the restriction endonuclease digestion sites is called as the hypervariable regions between individuals within a population. which is caused by mutation, rearrangement, deletion and unequal crossover. These small differences give rise to Rflp, microsatellites and a VNTR, and can be detected by using VNTR, and can be detected by separating the DNA fragments using the gel electrophoresis. Variants of Rflp are now used to overcome the limitation of the Rflps. T-Rflp is one such variant that is used to detect variants among microbes. Thank you.